Hello, everyone. Welcome to this presentation. My name is Jorge Castillo, and I'm substituting Professor Abadia, who is unable to attend this talk for personal reasons. We would like to thank the organizers for giving us the opportunity to present our recent work on zinc deficiency in Prunus rootstock GF677. And I'd like to start this talk giving you some background information. The transition metal zinc is an essential micronutrient for plant growth, and this metal shows some physical and chemical features like redox stability under physiological conditions, a high tendency to bind ligands with nitrogen, oxygen, and sulfur donors, and is the only metal able to adopt tetrahedral coordination geometries. These unique features make this metal an ideal ion to participate in several plant functions. For example, photosynthesis, carbohydrate metabolism, membrane stabilization, protein synthesis, enzymatic activity, DNA and RNA regulation, and biotic stress responses. For several proteins, zinc plays roles as structural catalytic cofactor. And this is the only metal present in enzymes of all 6 classes. Although zinc is very important for plant growth, zinc deficiency is a widespread micronutrient disorder affecting crop production in agricultural soils. This is in part because the soluble soil zinc fraction available for root uptake is decreased by several factors, including alkaline pH, redox potential, high contents of bicarbonates, organic matter, and phosphorus. Plants grown under these conditions usually show leaf chlorosis, reduced leaf size, and stunted growth, as well as decreases in photosynthesis rates and decreases in the activity of the carbonic anhydrase. This enzyme catalyzes the conversion of carbon dioxide to carbonic acid and facilitates the diffusion of carbon for photosynthesis. Due to the low levels of zinc in soils of available zinc, plants have evolved several response mechanisms to overcome the zinc deficiency effects and to maintain optimal concentrations of zinc in tissues. These mechanisms include the coordination of zinc transporters and synthesis of molecular ligands like amino acids and carboxylates in order to enhance the zinc uptake and the translocation from roots to shoots. In this work, we describe the main physiological changes and root responses induced by zinc deficiency in the Prunus rootstock GF677, with the aim to develop further detailed studies with this system on plant zinc homeostasis. For this purpose, we obtain plants growing peat from an agromillora nursery. These plants were hydroponic adapted in a growth chamber under controlled conditions. For these adaptations, plants were transferred to a 10-liter container filled with a diluted Hoagland solution at pH 5.5. The concentration of this nutrient solution was increased every 2 or 3 days until reach a half concentrated Hoagland solution and pH 5.5. Then, for the treatment imposition, plants were transferred to 4 little buckets containing the same Hoagland half concentrated solution at pH 5.5. 4 plants per bucket were grown with 2.4 micromolar of zinc for control plants, and the zinc deficient plants were grown in the absence of this nutrient. Along the period of time, spot measures were taken and plants were sampled at 3 established dates, 7, 14, and 21 days after the treatment onset. In these sampling dates, plants were separated in apical, young, and old leaves and roots for mineral, pigments, enzymatic, and metabolites analysis. Several batches of plants were run to collect leaf and roots. Now we move on the results section. Here in these plots, the zinc control plants are represented in green, and the zinc deficient plants are represented in red. Here in this plot and in the followings. Well, the zinc deficiency was monitored with the zinc concentration in tissues, and here when compared the zinc concentrations of control plants with zinc deficient plants, we found that the zinc concentration was decreased in all tissues tested. The decreases were in the range from 37% in apical leaves at day 7 to 92% in roots at day 21. Zinc deficiency was also monitored with the activity of the anhydrous carbonic anhydride in extracts of young leaves. Here we found that the activity of this enzyme was also decreased with time. The percentage activities of carbonic anhydride in zinc deficient extracts were in the range of 50% at day 7 and 30% at day 21. After the zinc deficiency was confirmed, we look for changes in other micronutrients concentrations in tissues, and we found that the concentration of iron and manganese increased in young and old leaves, whereas the concentration of copper decreased in young leaves. Concentrations of these micronutrients in apical leaves and in roots were not affected by the zinc deficiency treatment. Regarding to macronutrients, a similar effect was found. Here, the calcium concentration increased in young and old leaves, whereas decreased in the roots. Magnesium was only affected at day 21 in old leaves, and potassium increased its concentration with time in old leaves, and in young leaves only the first sampling date. This picture shows the apical and young leaves of plants sampled at the end of the experiment, 21 days after the treatment onset. And here we can clearly see that the zinc deficiency led to chlorosis in these leaves. In the next plot, we show the timing course of the spot values, and significant decreases were found from day 4 to the end of the experiment in young leaves. This effect was found in apical leaves since day 9 and to the end of the experiment. The decrease ranges were between 5 and 16% for young leaves and 12 to 31% for apical leaves. When we analyzed 8 photosynthetic pigments at day 21, we found that the concentrations of all of them were reduced in apical and young leaves. These decreases were more intense in apical leaves than in the young ones. The range of decreases were between 53% for biolaxantine pigment cycles to 69% for chlorophyll B. Now, this picture showed the root of a zinc deficient plant grown 21 days in these conditions. And in extracts of this tissue, we can find 25 metabolites were detected. This Venn diagram show the metabolites decreasing, the metabolites increasing, and metabolites that show no changes. One carboxylate, 2 sugars, and 2 amino acids show concentration decreases, whereas 1 amino acid and 1 alkaloid slightly increase in the zinc deficient roots. The larger decrease were for the carboxylate citrate with 43% of its concentration decreased. Well, now let me summarize the main points. Zinc deficiency causes decreases in zinc concentration in all tissues and in the carbonic anhydride activity in young leaves of the Prunus rootstock GF677. Zinc deficiency also caused decreases in the photosynthetic pigments in young and apical leaves. Also, alteration in the plant ionome with increases of iron, magnesium, calcium, and potassium in old and young leaves, and decreases of copper in old leaves and calcium in roots. And alteration of the root metabolome with decreases in citrate, sucrose, trehalose, glutamate, and glycine, and slight increases in arginine and trigonelline. This work was carried out at the Aula Dei experimental station in Zaragoza, Spain. And finally, I want to thank the financial support of the Spanish State Research Agency and the Mexican National Council of Science and Technology. Thank you very much for your attention, and that is all.